All right. Good morning, afternoon, evening, uh, everyone. Thank you again for joining us this week. Uh, this week we are doing amplicon sequencing analysis. Um, Most people, when they think of this, they think 16S, which is the majority of it, but these same processes can be applied to ITS sequencing for fungi, for example, um, and other sorts of marker gene analyses. Okay. All right. So, familiar slide by now. We are here in week 3. And an update. We are swapping out weeks 4. So next week was originally supposed to be Dr. Ahmed Zaid talking about advanced ecological statistics. We are swapping that for the final lecture with Dr. Olivier Zablocki. Hopefully I'm pronouncing that correctly. Apologies if not. So next week, instead of advanced ecological statistics, we will be discussing metagenomics with hybrid short and long read sequencing technologies. So for example, combining nanopore sequencing with Illumina sequencing. So look for that next week instead of the originally scheduled ecological statistics. Okay. So just a little bit of background. Community ecology is really what we're trying to do here. Now, whether we're counting bears and tigers or counting microbes, ecology is the scientific understanding of factors determining the abundance and distribution of species. And that's the sort of core thing that we're interested in. So individual responses to biotic or abiotic environments. So for example, one of the things I work on is how oral microbiota react to titanium dental implants, for example, or ceramic dental implants, which is a form of abiotic surface or environment. How species interact with each other, either metabolically or antagonistically. And so community ecology then is trying to get an integrative understanding of how all the different factors of a local environment mold local species pools at different scales. So for example, one of these things that we're interested in is assembly processes. How do communities form, right? Again, to go back to my particular research, you get a dental implant or a baby is born, right? And they have as soon as they're born, they're introduced to all the different microbiota in the environment from their mother, etc. So in new environments, how do species assemble and how do microbiomes form? This is super important for understanding how communities can be resilient to ecological disasters. How do those communities reform? And it can be also important to, for example, in human health, an ecological disaster is disease, right? So how do repeated insults with disease? In my area of research, for example, gingivitis is a reversible disease condition, right? But over so many repetitions of gingivitis, in some people it can turn into gum disease or periodontitis. So where is community assembly and adaptation playing into that? How do species traits uh, interact with environmental insults to change species assembly, species or community composition. So these are all the sort of large-scale things, sort of in one figure there. And so these are some of the key questions in community ecology. And so you've got butterflies of different colors there. The green and white colors represent differences in environmental conditions, and the butterflies with different colors represent different species. And the boxes represent different sampling units, right? So you go, if you want to go to oceans, right? We take a sample from here. We go 5 miles on our boat and select another sample, right? So we have different samples. Or again, going back to my particular area of research, we sample different teeth. So we ask how species occurrence data can be used to understand how the processes in different environmental conditions affect community structure. How can we predict communities under new conditions? For example, the question mark box there. Some species are found to co-occur more or less often. So again, these are the high-level questions we need to ask before even getting into any particular study of a certain microbial environment or microbial ecosystem. This is just a particularly useful quote I found that's useful again for kind of distilling all this down into one sentence. So the important elements for analysis include species fluxes, interactions, organismal properties, functional, reactive, analysis of functional pathways. We want to understand how communities react to nutrients in their environment. How does that change when external events happen? So for example, one of the things we just published a paper on e-cigarettes. So how does the aerosol in e-cigarettes affect the microbiome in the mouth? And for example, it turns out that the liquid in e-cigarettes is comprised mostly of vegetable glycerin and propylene glycol, which are both simple 3-carbon molecules. And it turns out that bacteria really like simple 3-carbon molecules. And so how do communities change when they have an abundance of nutrients or have severely restricted nutrients? And how does that, one, change individual organisms? And how do those shifts affect interactions among organisms? How do organisms interact generally? And so some of the emergent properties of these questions are taxonomic diversity, functional diversity, functional redundancy. As new taxa are added to an ecosystem, the number of ecological functions that are present increase, but at some point additional taxa don't add novel capacities. They do add redundancy to specific functions. And so in general, highly redundant systems are more likely to retain a function after environmental stress because the probability of an organism containing those functional properties remaining after the stress is greater. Resistance, for example, is the degree that a system does not exhibit an acute loss of function after a stress is placed on a community, and resilience is the rate at which functionality returns or taxa return after an acute stress. Now, with amplicon studies that we're going to talk about here, the analysis of functional diversity, functional pathways, also is not really possible because we're just looking at organismal markers. But we can look at, there are methods for analyzing genetic potential. So things like PyCrust, for example, tools like PyCrust can say, well, hey, you've got all these taxa, and we know these organisms have these annotations, and so we can kind of guess what a metagenomic study might look like just from the numbers of organisms that are present. So this figure is definitely starting to show its age, but for this talk it's useful. This is a sort of general overview from start to finish of the kinds of analyses that we're talking about, from sampling all the way through to different sorts of analyses. So why marker genes, especially now? You know, we've got the 10x genomics happening, the NovaSeq, all of so the high throughput and the long length. So why are we still looking at marker gene analysis? But first, why marker genes? So first, marker genes are required to be ubiquitous. They have a mix of highly conserved and hypervariable regions. And the hypervariable regions is what allows us to differentiate between organisms. So common marker genes for bacteria and archaea is the 16S ribosomal subunit, and fungi, the 18S or the ITS regions, also ribosomal RNA. So as to why, again, marker gene analysis is cheap and scalable, although both of these are becoming somewhat less of an issue, especially in terms of cost. But in terms of depth of sequencing for the kind of taxonomic identification that you're getting, 16S analysis still, or marker gene analysis on something like the Illumina MySeq is still very useful for initial analyses of new systems that people haven't studied yet. So this is just sort of a cool picture of all the tools, little, what's the word we're looking for, noteboard kind of layout of some of the different tools that people use for this kind of analysis, just to give everyone a general idea. And this is a very small selection of all the tools that are available. And this actually brings up an important point that I do want to make, that this is also applies to the rest of the webinar, that these are very large areas of research and very large areas of work that have been going on for quite a while now. And so there are hundreds of tools and new tools being published every day. And it can be hard to keep up, but no one can keep up with everything. We try our best. And there are typically a consensus of tools that people use, so don't feel behind if you're not using the latest, greatest, bleeding edge. But at the same time, that's available. Once we learn the basics, once we learn the standards, then it becomes easier to incorporate new tools into your knowledge bank as time goes on. So just to reiterate and summarize, don't be overwhelmed by all of the different tools that we're presenting, or by uh, the Google Scholar feed you may be getting, um, or whatever journal email list you may have signed up for. Um, like I said, there's typically consensus for all types of analyses. Learn that, and then add new things as time goes on. So this is sort of the general, very general, and even the arrows aren't necessarily uh, always the same depending on what tool set you're using. But in general, we start with raw data. We do some sort of uh, trimming and filtering, quality correction. And if you're working with paired end data, then you have to merge the paired ends. And then we go on to either OTU clustering or ASV construction, which we'll get to those later. Removal of chimeras, and again, the order of this can be different for different tools. Constructing phylogenetic trees for analysis, taxonomy assignment, and then all the different methods down below. So I do have to mention OTUs versus ASVs. So OTUs, as you can see in that first paper there, stands for Operational Taxonomic Units. It came about with some of the first approaches for analyzing marker gene data, which was similarity clustering. You can see that in the second paper there from Robert Edgar, the famous QIIME, uparse, unoise tools. But the OTU is a stand-in for a biological, some biological unit, whether that's species or something higher. But when you do de novo sequencing, as they call it, is you're taking raw sequences and without reference to some database, grouping the sequences by some similarity. Classically, that was 97%. That if two sequences were different by no more than 3%, they could be considered the same organism, whether that's again species or some higher level. But since it was done without reference to some database, and we were just clustering these sequences based on sequence similarity, then the term operational taxonomic unit came about as sort of the precursor to taxonomic assignment. As we know, these clusters at 97% or more represent potentially some organism, but we don't know what it is yet. And about 2015-ish, plus or minus a couple years, depending on the tool, people started coming out with new kinds of tools that sort of dug a little deeper into what's possible with marker gene analysis. And so, as you see in the first paper there, these were called exact sequence variants or amplicon sequence variants. There's a whole 30-plus page discussion on, I believe it's the DADA2 GitHub site. Someone posted about, hey, there's all these methods like DADA2, and what should we call them? And so you can go find that on GitHub. But anyway, it's all referencing the same sort of thing, that instead of just clustering by sequence similarity, these tools go a little bit further and try to model the error in sequencing. So with Illumina sequencing or any of these sequencing by synthesis, there are always errors in base calling that creep in. We can try to control those errors with quality scores, but some of these tools go a little bit further and say, hey, maybe we can model the errors and have an expected sort of transition diagram. How often will an A be called a C or a G be called a T? Those sorts of things. and try to model the error and then correct that error. And in doing so, you sort of get a side benefit of being able to cluster at a single nucleotide level. And so, as I said, that first paper, exact sequence variance paper, is sort of a good introduction to the idea of exact sequence variance, or ASVs. So it's a good read for a sort of general introduction. Now, as I said, there's a whole bunch of different tools. DADA2 is one of them. UNoise3, dblur, MED, minimum entropy decomposition. So there's a whole bunch of different tools for doing this sort of approach. Oligotyping, swarm. There's a number of these different tools. So the question is, well, which tool do we use? This is a good paper, one of the first to actually look at this question, with a great title, of course. And they went and compared DADA2, UNoise3, and dblur to the standard uclust approach for OTU clustering. So their conclusion was that despite the differences between all the underlying algorithms, that all four approaches were generally comparable based on weighted unifrac and Bray-Curtis distances, which we'll talk about what those mean later, but sort of general community composition similarity. But the tools vary greatly when looking at unweighted or raw numbers of ASVs or OTUs. With the older open de novo clustering approach of uclust, generating many more OTUs than the ASV approaches, many of which aren't really real or add false diversity. And so they said in all cases the open reference OTU clustering or the de novo OTU clustering had greatly increased numbers of OTUs when compared to the ASVs found by the denoising pipelines. So we're going to get into doing some of this. We're going to start with DADA2, mainly because the implementation of DADA2 in QIIME misses a lot of stuff, which if you remember last week when we talked about why graphical interfaces haven't won out over text-based Unix interfaces, it's for the same reason. Anytime you try to implement an interface to something else, there's always complexities and things that you miss. And two, since DADA2 is implemented in R, you can actually do the whole, with a combination of R and Phyloseq and other libraries, you can actually do your entire marker gene analysis just in R, which is appealing to some people. So let us switch over. So if you looked at the webinar resources page, so I will switch over to that now. Okay, let's see. Okay. All right. Is my browser window showing up here on the screen?
Yes.
Okay, thank you. Yeah, sometimes I've noticed I've got multiple screens, like I said last week, and so sometimes moving back and forth between things can be a problem. But anyway, so this is the webinar resources page. So if you haven't had a chance, these are the R libraries and software that we'll be using, as well as links to download all of the test data and taxonomy training for the different tools that we'll be using. Let's see, I think I need to refresh this. Yes, there we go. And so as well, there's I've put up a GitHub repository with all of the step-by-step analyses in R or at the command line for QIIME and Mothur. So this is what we're going to be going through. This is all in R. So base R doesn't have a really great interface, but R Studio is an excellent tool, and so I've switched to that now. Let me clean this here. I was just running things earlier. So I'm just going to go ahead and just terminate everything so that we start with a clean environment. And actually I need to switch ahead for a minute just to, let's see, do I have the data? Let's see. Okay. Yeah. Okay. Seem to have forgotten a slide here. I just wanted to sort of introduce the data. So this is, let me bring it up here. Okay. So this data is from is actually borrowed from Mothur, and specifically Pat Schloss's, the author of Mothur, his lab's MySeq standard operating procedure. So this is the data he has available, and it's on the resources page to download. The standard Mothur and so both the standard Mothur tutorial and the standard DADA2 tutorial use this data, but they use so this is feeding mice. So they have 6 female mice and 6 male mice. They track them over about 150 days. Some of them get and they're just allowed to eat. And they looked at differences in weight gain and how that relates to the microbiome. So this is just sort of a standard data set that people use. It's relatively small. It's and particularly for the standard Mothur tutorial and the standard DADA2 tutorial, they use 20 samples from one single female mouse through the study. I want to do things a little bit differently, so I took a subset. So you can download the full data set, several gigabytes, but I created a subset that is simply days 0 and days 149 for all the male and female mice. So like I said, that data can be downloaded from the webinar resources page. The Dropbox link here has the mouse gut data. So we'll be using that same data set for all 3 approaches, DADA2, QIIME, and Mothur. So we'll use that same mouse gut data as we go along. Also, I just realized I have not been tracking the Q&A. I haven't heard anything yet. I guess there's no Q&A. There we go. Okay. Yes. There's apparently some technical issue with Zoom, so we don't have the standard Q&A like we did last year.
Someone's asking if you can share the GitHub link. That would be really helpful.
Yep. So I said again, all of this is on the, okay, yeah, there we go. Someone just pasted it. And I'm seeing a question about differences between when to use OTUs versus ASVs. So in general, most people have moved to using ASVs, with the notable exception of Mothur. If we go back to uh, this paper here, the updating the 97% identity. Um, when this was originally put up on bioRxiv as a preprint, which allows commenting, Patch Sloss posted some very lengthy comments about his opinion on ASVs, and that he thinks they have some issues. Um, so, for example, we know that organisms can have multiple copies of the 16S gene, but some organisms can even have differences, sequence level differences in those genes within the same genome. Um, and so he was concerned that the ASV methods with their single nucleotide accuracy could potentially split those up into multiple organisms, um, uh, and create problems there. There have been some reviews since then that have shown that's generally not too much of an issue. But some other Mothur, however, has not stood still. They've implemented some more recent updated versions of OTU clustering that we'll get to, but the version that's recommended in Mothur is called OptiClust, and he uses the Matthews correlation coefficient to update those clusters in sort of an iterative cycle. However, I will say, and we'll get to this in Mothur, but Mothur does implement a sort of ASV kind of thing. The default is a version of single linkage clustering, but they do re-implement a version of deblur, which is the ASV method that the QIIME2 authors put together. So, in terms of OTUs versus ASVs, in general, OTU methods generate more clusters than ASVs. ASVs can often be more accurate, but it really in a lot of ways depends on your particular microbiome of study, the complexity of that microbiome, the characteristics, what kind of organisms are in the microbiome. So again, going back to the idea of organisms having multiple 16S genes that can differ by multiple nucleotides. So the answer is there isn't any one answer, as with a lot of these kinds of things. But in general, people have moved mostly to the ASVs. But that doesn't mean that updated versions of the standard or what we used to call OTU clustering isn't still applicable. Yes. Okay. Yes, and Sarah, yeah. Apologies, things were kind of I got things up a little bit late. I was trying to get things up a little bit earlier, but unfortunately that didn't happen. But at the same time, all this stuff can be downloaded pretty quickly or installed relatively easily. So we can do that as we go along. Okay. So I'm seeing a question about tools and programs working on Windows. So that is one of the nice things about Mothur, is that they write almost everything themselves in C++. And so Mothur works equally well on Windows, Mac OS, and Linux. The other tools, not so much. So QIIME and DADA2 R libraries in general don't really work great in Windows, and a lot of these tools, don't even work at all in Windows. So that can be an issue. But like we discussed last week, there are you can get virtual machines, so you can use a tool like VirtualBox to install Linux to run Linux inside of your Windows machine if you don't have access to anything like that, or Docker, as someone said. Yeah, so DADA2 can be installed in Windows. Someone's saying, but a lot of the ecological analysis tools, a lot of the libraries for ecological analysis may or may not work. And the Windows subsystem for Linux, I don't know how many people have had success with that. I've heard mixed things. So I'm not saying it's impossible to get it running on Windows, but it's certainly going to be a lot more difficult. So what is specifically about the QIIME implementation of DADA2? So for one thing, they don't implement pooling, which we'll discuss, which is the method that is implemented in DADA2 for picking up on the rarer organisms by sharing information between samples. So normally in DADA2, samples are processed individually, and something isn't considered an ASV unless it's present more than once. So by sharing information between samples, it can pick up on some of the more rare sequences that only occur in a few samples. But if you were to process things individually, then it wouldn't be considered as an ASV. So there are some other things. Mainly, my comment there, other than the issue of pooling, comes specifically from Ben Callahan, who said that the QIIME implementation was missing some things. So that's mainly where I'm saying that from. Okay. All right. Okay. So let's get back to, yes, okay. So this is the original paper for DADA2. So let's switch over to actually running things. So this, we'll see how this goes. Some of these steps take a little bit more time. Nothing should take more than about a minute to a minute and a half. So we'll see how things go. So in R, again, you don't have to, but in general, it's a good idea to load all of your libraries at the beginning. But again, we'll see later. I don't always. This is not always followed, but just helps for readability. So the first thing that we're going to do, also, I want to point out the library here. The talk from Jenny Bryan that I mentioned last week. She's a huge fan of the here library. Basically, it's an alternative to using the setwd, the set working directory command in R, which is very fragile. So if you set your working directory, it's entirely dependent on where your data exists. Okay, so where can I access the R script? The R script is on the GitHub repository. I can link to it directly. Let's see. Yeah, okay. There's the direct link to the R script. So for this, if you, I have a folder for DADA2 that has this script in it, plus a folder containing all of the raw data, which I can link here. So if you download this data set and put it in a folder, extract it in the same folder as the R file, and call it data, then you should be good to go. And so you'll see here, so I'm using here you'll see that when you load the here library, it tells you where it considers here to be. This is just a path on my hard drive, but here uses some heuristics. So it typically looks for our project files. So RStudio, you can create projects, and so if here finds a project file, then it will say, okay, that's where we are now. If not, then it typically will start from your home directory. But either way, it provides sort of predictable heuristics for locating yourself, that so you can avoid hard coding specific paths. And so this is a good example, right? All of you are potentially following along with me, but you are not inside of my computer. So my path to the folder, to the data folder, is going to be very different from your path to the data folder. But with appropriate use of here, we can sort of bypass a lot of that and have everyone, especially for sharing, if you're working with multiple people working on projects, here is a much better tool to use than sitwd. So the first thing that we're going to do is just retrieve the sequence paths. So we're constructing a path. So list.files just goes out to your hard drive and pulls out a list of files and returns it as an actual list in R. And so you just give it a path, which is what we're using for here. You give it an optional pattern of files. So here we want to collect the forward reads, so R1. And let me check to make sure. Okay, yeah. Let me clear this here. Okay. Okay. These are gzip, so we'll need to edit that. And so then we can look at forward FP. So I'll switch back and forth between running lines in the R file and doing some output checking in the console below. So I'll be switching back and forth between those. Hopefully the font sizes are okay for people. If not, you can hopefully follow along with the R code. So we can take a look at the first thing. So the first file it found was F3D0, so female mouse 3 at day 0. And we can do the same for the reverse. So again, rev FPs. So again, it finds the same, but the R2 instead of R1. So we start off with a separated list of forward and reverse reads, and then from the excellent string R library, there's this stir split n function. This in particular is actually, even though it's on the website, on their documentation, I found out that you have to install the development version of string R. It's not in the main release yet. So if you don't have this, there's another way to do it, but this is by far the easiest way to take the sample files and extract sample IDs from them. So what we're doing here is base name is a vectorized function that goes through each of these paths and extracts just the file name, so everything after the final /. So it starts, so we get just the file name, and then stir split n splits up that name by underscores and returns just the first item after splitting, which in this case the standard Illumina file names the sample name is the first thing before any of the underscores. So what we're really doing is just extracting that. And so we can see, so well, let's run this first. Sample IDs, and then so we can say oops, sample IDs and take a look. And so that's our vector of sample IDs. So you can see here we have female 3, day 0, female 3, day 149, female mouse 4, day 0, 149, etc., etc. And then all of the samples that start with M. This is male mouse 1, day 0, male mouse 1, day 49. So you can see all the different samples that we have there, and the sort of pattern. And we'll use the construction, the standard construction of these sample IDs later on to develop a metadata table. And I just want to point out, this is kind of what I was talking about last week with naming things, but especially sample names. If you have multiple groupings to your sample names, please don't use underscores just because of the standard Illumina naming by separating sample ID, sample number, lane, read, etc. by underscores. So if you do need to separate your sample IDs into groups, use a dash or something else. It'll make your life a lot easier. Okay, so we extracted the sample IDs from the file names. So the first thing we want to do is sequence trimming. So DADA2 has a plot quality profile function that basically just takes a random sample from the file you give it. Here I've had it do it for the first 4 samples. You can see the sample name at the top there. And so this is the sample quality graph. Yeah. So any mistakes that I correct in here will be updated after the webinar in that GitHub page. So you can just and you can see in GitHub when changes are made, what changes were made. So you can just keep checking back for any changes there. Okay. Yeah. So this just shows for 4 of the files, for some number of reads. For random sampling of reads, what the general quality profiles of those are. The green here shows sort of the average quality score for all of those reads, and then the orange lines show the top and bottom quartiles for those. So you can see here, so these are 2 by 250 MySeq reads. And it's one of the issues with the Illumina platform in general, that as you get closer to the end of the read, the quality can drop off. In the forward reads, it's not so bad, but in the reverse reads, reads in Illumina can have very poor quality. So it's definitely important to check that out. In general, before this, I would recommend using a tool like FastQC and MultiQC to check those. If we have time, we might be able to go through that. But for now, we can use the built-in quality checking tool for DADA2. But you can see, so around 240 or so, the quality starts to drop a little bit. In general, anything above 30 is very good quality. Some people say 35. Anything above 20 is good quality. Anything below 20, and then you're really starting to get iffy. The nice thing about the paired end sequencing is that the reverse or the end of the since we're reverse complementing to merge, the poor quality reads in the reverse can be sort of rescued by the much better quality reads at the end of the first read. But we'll need to do some trimming to help things out. And this is one of the reasons that so this is sequenced on the V4 region of the 16S gene, um, uh, which is sort of the standard for a lot of 16S sequencing, mainly because um, it's about 250 base pairs, so you can get full coverage, more or less full coverage from both ends on the MiSeq using the 2x250 reads. So it's a lot of people use that, and this study also used that V4 region. So we can trim a whole bunch off of the reverse read and still have substantial overlap. So we don't necessarily need to worry about quality drop-off. So we can see here, so that was the forward reads, and we can look at the quality for the reverse reads. And so you can see here what I was talking about. So at around 160 base pairs or so, depending on the read, if we look at the green, so the orange can kind of distract things a little bit. So the average reads are still pretty good, out to about 200. They're still above 20, but that's the average. So depending on the sequence, you may have better or worse quality going back to about 160. And so that's where we'll end up trimming on the reverse read. So we use those graphs to sort of guide our filtering for later. Okay. So the next thing we'll do is set up our filtering paths. So we're just creating new paths that will be used by the filter function. These are basically just output file names. So we can look at that. So we're literally creating a new path to a folder called filtered. If this folder doesn't exist, then the filter and trim function will create it. And we're constructing a string consisting of the sample ID followed by _f for the forward reads and _r for the reverse reads, _filt to indicate that it was filtered in the FASTQ GC. Yeah. So someone's asking about paired end 150 base pair reads if they don't overlap. Yeah. So using the forward reads in that case is often recommended if you've got really poor quality data that won't overlap. If the data won't overlap, there's not much you can do about it. And so going ahead using just the forward reads, since the forward reads typically have much better quality than the reverse reads, is generally the way to go with that. It's unfortunate, but it's better than nothing. Okay. Yeah, and if you look at some of the QIIME, there's multiple QIIME 2 tutorials with different data sets, and they go through some of that. So yeah, that can be an issue definitely with the reads not overlapping. You can also, in some cases, if there could be an overlap, you can also sort of relax a little bit on the quality filtering. You don't want to relax too much, but depending on the quality profile, if you can relax things just a little bit, then you might be able to get some merging, but it will really depend on your data. So this is the filter and trim function. Before I do that, I'll point this out. The TikTok library is not for generating funny short videos from your R code. It is for function timing. So R does have a system.time, I believe, some sort of function that you can wrap around a single line. But if you want to profile multiple lines, then the TikTok library is very, very useful. You simply call tick where you're starting, and at the end of your code that you want to profile, you call talk, and then it automatically calculates how much time it took to do that. Now, the standard DADA2 tutorial includes this max EE parameter, which is sort of the max expected errors. And they use the standard quality formatting, or sorry, quality formula for base calling. So their standard is approximately two expected errors for the forward and reverse reads. There was a recent paper from last year in PLOS One. It's called Comparing Bioinformatic Pipelines for Microbial 16S RNA amplicon Sequencing. I will paste the link into the chat. And so they recommended actually that the max EE parameter can cause some issues with DADA2, and so they actually recommended leaving it off. Yeah, I just came across this. This is fairly new, so run it with, run it without. That's the nice thing. You can check with this reduced data set. It doesn't take too long to play around with it, but yeah, go ahead and check out that paper. So we'll leave it off for now. Okay, yeah, so what does the TikTok output line look like? We will run it now, and you will be able to see. Let me clear this up here. Okay. All right. So we're going to run. This shouldn't take too long to do all the filtering. But so you'll notice again that so the trunk len here is the forward and reverse. So we want to keep the first 240 base pairs on the forward read and the first 160 base pairs on the reverse read. And then it will compress the output, so all the output files will be gzipped as well. One caveat, that the DADA2 tutorial does mention that on Windows, this multi-thread should be set to false. Okay, so if you look into the console down below, this is the output. It's just the number of seconds elapsed from the command that it was wrapped around. Okay, so now we have filtered and trimmed data files, and we're going to start building a data table or a tibble in the tidyverse with the that will use this to track the number of reads that are present at each stage as we go through different filtering options. Yes, so thank you. So someone says, do you need to trim adapters before DADA2? Yes. In this case, the data we're using, just to save some time, this data doesn't have any linkers or adapters or primers or anything like that. It's already been trimmed. But yes, the required pre-processing for DADA2 is to remove any linker, primer, or adapter sequences. Yeah. Okay. Yeah. Okay. So we're just going to construct a data table here. The first column is going to be the sample IDs. The second column is going to be the number of input sequences. And the third column will be the number of sequences following filtering. So we'll construct that table. Okay, so yeah, so for trimming adapters, the sort of gold standard for trimming adapters is a tool called QuitAdapt. That's pretty much everyone uses QuitAdapt for trimming. Yeah, here we paste the link for that. Okay. All right. Yeah, that's very easy to use. You just give it your, especially since the adapters tend to come before the primers, you can just give it the primers and it'll cut off everything from the primers and before. So it's very easy to use and is very adaptable. Okay, so we created our filter trim results table, or tibble, and we can take a look at it now. So here's our tibble, 24 samples by 3 rows. So again, all the sample IDs. Oops, that is supposed to be sample IDs, not the. Yeah, I need to change that. That should be sample IDs. There we go. Okay, so let's reconstruct that and take another look. There we go. Okay. So we can see that we didn't lose too many sequences from each of the samples. So in general, they're, as we said, they're pretty good quality. So we can go on. So learning errors. This is the error modeling step that's key to DADA2's version of ASVs. And so they read in a certain number of, so DADA2, the learn errors, reads in a certain number of reads until it gets to about 1 million base pairs, or 1 million bases, to do its error modeling, so that it can later correct potential errors based on quality data, so it can generate ASVs. So we will run that for the forward reads with TikTok, so we can see it shouldn't take too long. So you can see there the output. So it tells you how many reads it read in, how many total bases it's using. I think 1 million might have been, that may have been older. They may have updated that. But the general idea is they read in a chunk of reads from your data. Now, one thing I will say that's nice about DADA2 is that if you have the same person doing your library preparation, and the sequencing is done on the same machine by the same operator, then, and typically he recommends this for sequencing centers, for example. But you know, again, assuming a little error in the preparation steps for the sequencing, you can actually use the error model. You can save the error model and reuse it. But at the same time, it's a pretty quick process. So unless you're dealing with thousands of samples, then it's probably better just to redo the error learning. So while that is running, okay, yeah. So once we finish generating the error profiles, there's a way you can check how well the error profiles, the learned error rates fit the actual data. It's not really necessary. I haven't really come across in my use of the tool any particular any case where the error learning step didn't really fit well. So, but he does recommend that if you do notice something weird in the plot errors, that he recommends going back to learn errors function, and one of the parameters for it. Let's see. So the n bases. Okay, sorry. I guess it was. Yeah. So the default number of bases is 10 to the 8th. Yeah. So he deprecated the use of number of reads and defaults to the total number of bases. So 10 to the 8th bases, and however many reads it takes to get there. So if you do have an issue with your errors, with your modeled errors not fitting the actual errors, then you can increase this basically just to get more data. So Okay, this is taking a little bit longer than it did when I was running it earlier, so we'll wait for that. So the next step following learning the errors is the ASV inference or sequence correction. So going through all of the reads and incorporating the learned error model along with the quality data that's included in the FASTQ files to try to actually correct at the base level possible base calling errors so that the grouping of sequences can get down to ideally single nucleotide resolution, which is the sort of the definition of the amplicon sequence variance. So this DADA function is sort of the core of the ASV inference, and so we run the ASV generation on the reads separately, and then following the generation of ASVs, we will the ASVs will be merged. So you do your merging, which is a little bit different from most of the other tools. So both Mothur and dblur do paired end merging before they do their OTU or ASV clustering steps. So we will run that, and if you'll notice here, so we're giving it the filtered files, the error model, and this isn't in the standard tutorial, but this is what I mentioned earlier about sequence pooling. So it does some information sharing. So the options here are none, or no pooling, or pseudo pooling, which sort of well, let's see. So the full pooling actually processes all the samples together, which takes a substantial amount of time. The pseudo pooling runs everything individually, and then after the fact does some information sharing to try to find low abundance sequences. So this is sort of a happy medium, both in terms of running time and in not finding in finding fewer spurious ASVs. So we will run that, and there's a whole page on the DADA2 website about pooling and the various pros and cons and how it actually works. If anyone's interested in actually reading through that, and while that's running, I will actually go and find that and paste it into the chat so that people can take a look at that. Let's see. Here we go. Yes. Okay. Yeah. So here's the explanation for pooling that DADA2 uses. Okay. So you can see here that it ran through and generated ASVs for each of the samples separately, and then step 2, it goes back and like I said, does the sort of shared, the pseudo pooling. Okay, and then we will do the same for the reverse reads. Okay, while it's running. So that's running, so we can look at, okay, so as I said, after the ASV generation, it takes the ASVs generated for each of the reads individually or separately, and does the paired end merging on the ASVs instead of on the filtered data like most other approaches take. So, okay, so that's done. So we can take a look at what's in the ASVs. So there's one of these. You'll notice that this is just the first data, the data forward reads object is an array. So this is the first sample. So the first sample found 199 ASVs from 2,400 unique input sequences. And you can see the key parameters here. This is obviously way outside of the scope of this talk, but there are parameters that you can modify if you want to. So, okay. And you can also use this help to get a better idea of what's in the DADA class objects if you want to look at them for other things. Okay, so we will do the merge paired ends. And so it kind of gives you some nice information about how many pairs were successfully merged. And then at the end of this, we can take a look. That one's pretty fast. And so we can take a look at, so this is the first 6 ASVs, how abundant it was across the data sets, how many, and this is various parameters about the merged ASVs. So you can take a look at that. Okay, and then finally we can create our sequence table, which is just, this is sort of the OTU table. This is just ASVs as rows and samples as columns, and then you have the counts, how many of each ASV occur in each sample. And so we can look, and so the sequence table is 24. I'm sorry, it's yeah, I had that backwards. So we can just take a look at it here. Seek tab. Yeah. So it's actually yeah, samples as rows and ASVs as columns. But so we can see it. This ASV occurs 592 times, and female 3, day 0, etc. So okay. All right. Okay. So the next thing we want to do is get a look at the distribution of sequence lengths, ASV lengths. So we know our V4 region is about 250 base pairs. So these hypervariable regions, the thing to remember about the hypervariable regions is it's not just, they're not hypervariable in just in the sequence content, but also in length. So when we say that the 16S gene is about 1,500 base pairs, the about part is important. And so there's definitely a distribution of lengths that you will find. So for the, this is pretty good. So our sequence lengths, our ASV sequence lengths vary from 251 to 255 base pairs. That's pretty good. That's not outside of what we would expect for some variation in length. But if you were out in like the 300 or 500 base pair range for some of these ASVs, those would not be real. And so, and this is what we'll do in this next step, is we'll actually, this isn't necessary for this particular data set, it's just to illustrate the purpose. If we don't want to keep anything outside of the range, things that are either too short or too long. So if we had ASVs that were 100 base pairs in length, those would be way too short. So we want to kind of slice out a range of ASVs in the expected length for the marker gene hypervariable region that we're looking at. So we can do that with this command here. And again, this was within the range, so it won't actually change anything. Now the last step here for processing in DADA2 is Chimera removal. And so this goes and looks and finds, and so it's removed by de novo. So this actually goes and looks, and you can read more about it on the website, but this actually goes and looks for ASVs that are exactly half of one ASV and half of another ASV, and identifies, and then we can remove those from the data set. Okay, so out of the 340 ASVs, identified 76 that it considered as chimeras. And removed them, so now we have 264 non-chimeric ASVs for a percentage of about 4-5% ASVs, chimeras. So in general, what I've noticed with my own data is on average, somewhere in the range of 5 to 10%. You can expect to be chimeras. A little bit past, if you get into 11, 12%, it's not terrible. If you're in the 30, 40, 50%, that could indicate some library prep or quality issues. But in general, you can expect to lose, like I said, in the 5 to 10% range of sequences to chimeras. And it's really just a feature of the whole PCR process and non-specific binding and that kind of thing in the cycling of the PCR machine. Okay. So, so now that we've sort of run through the processing pipeline for DADA2, we can take a look at how many reads were left after each step. So we're just defining a function here to get the unique reads at each stage. And so we're going to create another data table here that starts with the filter and trimming results that we generated early on. And then we're going to add additional columns to that. Forward denoising results, reverse denoising results, merged reads, and then finally the non-chimeric reads. So we can generate that table, and we can take a look at the head of it. So so this is our, again, the table we saw earlier. These first three columns are the table created earlier, and we added these columns. As you can see, so for example, for female 3, day 0, we start out with 7,700, almost 7,800 raw reads, and by the end of it, we'd lost about 800 reads. So that's not terrible. Again, it's about 10% over the whole thing. So some of these, and we can actually, we can view the entire table. As you can see, the whole table. And so some of these samples, so for like this sample, for example, is just poor quality from the beginning all the way through. So if you're starting with 83 reads, then you can't expect much. Again, this one, 14 reads, and by the end it had 0 reads left. So this mouse 3, day 149 was just something went wrong with it, and then it'll have to be removed essentially from the analysis. So, but in general, you're not losing. This is just a sanity check to make sure you're not losing a lot of sequences at any one particular step. Okay, so someone asks, so our interest in the tables in the last column? Yes and no. So the last column is important because that's what you'll be moving forward with in terms of analysis. But really, this is to, like I said, sanity check the process, right? So if you went from input to filtered, and you had 7,700 reads as input, and you ended up with 2,000 reads after filtering, then you might want to go back and look. Oh, did I make a typo? For example, did I instead of setting my trimming at 240 base pairs, did I leave off a 0 and accidentally make it 24 base pairs, right? So like I said, it's just a sanity check. So at each stage, you don't want to lose a giant proportion of your reads. If you do, then you should go back and check that step and see, was there a mistake made, or is there something wrong with the actual sequencing data itself? Okay. Is it necessary to normalize the data in order to have the same number of reads in each sample at the beginning? For this, for the data processing, no. Normalization is something that is important, or potentially important, depending on the kind of analysis you're doing when we get to the ecological statistics part. But for the data processing, the data is what it is. We're not doing any normalization at this stage. So, okay. All right, so that's pretty much it for the DADA2 processing. We want to get kind of moving here. So I'll just talk through the rest of this. If you want to, for example, run DADA2, but do the rest of your sequence, do the rest of your analysis in QIIME, for example, then you can export your ASVs, just your ASVs to a FASTA file in QIIME, and we'll see this in a minute here, but in QIIME this would be your, I'm blanking on the term, but basically you need a FASTA file. Your representative sequences is what they call it in QIIME. So your ASVs, QIIME does want a FASTA file of your ASVs. So this code will do that, generate that FASTA file. And then you can also export your sequence table, your counts table, to a biome table. Biome is sort of an open format biological observation matrix. Yes. So, okay, so to Nicholas, this whole process helps in analyzing raw data from the MySeq. Yes. So, yeah, so this, all three of these processes that we're going to go through, DADA2, QIIME, and Mothur, and we'll see how far we get in each one, is to take your raw sequence data and turn it into a table of counts that you can analyze. And whether those counts are ASV counts or taxonomy counts, you can do both, analyze both. Most of these tutorials we'll see will analyze, do your sum analysis at the ASV level, but ultimately ASVs don't mean anything until you assign taxonomy to them. So you can't make any ecological. There's a limit to the ecological statements you can make without knowing what organisms your ASVs are. So you'll need both at some point. But yeah, so this whole process is to take your raw sequence reads from the machine and turn it into something you can analyze qualitatively and statistically. Yeah, so dereplication happens automatically in DADA2. And Miguel's question about normalization, we'll have to come back to that later, or maybe you can email me or something. But that's getting a little bit ahead of ourselves. Yes. So ASVs are generated for the forward reads and the reverse reads separately. And then the merging is done on the ASVs. So like I said, that's sort of different from what QIIME, from what Diebler and Mothur do. They do the sequence merging, and then they do the ASV or generation or OTU clustering. So those are different. Okay. All right. Yeah, sorry if I can't answer all the questions. We'll be here for 5 hours. We're running a little bit behind. So like I said, so that's this section of code will let you export your DADA2 processing to QIIME or some other thing. Like I said, a biome table. QIIME uses biome tables, but it's not unique to QIIME. A biome table is just an open source format for storing biological observation data. So it can be used in a number of different tools to analyze your sequence counts. Now, like I said, this is just about clustering based on sequence similarity. And again, it's all the OTU ASV stuff, but that's I want to get into all the technicalities there. But so right now all we have is sequences. If you want to assign taxonomy, DADA2 implements a version of the naive Bayes clustering algorithm that was, I believe, first introduced by RDP, the Ribosomal Database Project. So the RDP classifier is basically being re-implemented in DADA2's assigned taxonomy. Now this only assigns taxonomy down to the genus level. So if you want species, then he does have an add species function here. But it's important to note that DADA2 only assigns, so this add species function only assigns a species label to an ASV if they are 100% identical down to the base level. There's a wide variety of opinions there. So the DADA2 tutorial mentions the ID taxa algorithm, which if you go read their paper, their authors say that 16S can never be used for species analysis, which, and again, I don't want to get into any sort of flame war here, but it depends on your data and your species. As a general statement, yeah, species can be hard to assign from 16S data, especially depending on the region you're looking at and the organism you're looking at. So the add species function here is fairly conservative and says the species only, an ASV is only this particular species if it matches 100%. So the QIIME2 implements its own version of taxonomy assignment using another naive Bayes classifier. It was trained somewhat differently. So they're a little bit more forgiving in terms of species analysis, species assignment. So again, it's really down to what you know about your data set, what you know about your environment and the organisms that are in it, as to how successful you'll be at assigning species. So for example, the strep species are notoriously difficult or notoriously similar at the species level. So you'll for a lot of those you'll only get genus level or higher assignments. But again, it's dependent on knowing your environment. So you can assign taxa, and then this is some code if you want to continue your analysis in R using the Phyloseq library. And many of the other libraries, Vegan, for example, provides a lot of the statistical analysis methods. So this whole section of code is just gathering data into a format that will let us create a Phyloseq object. And that Phyloseq object consists of an OTU table, sample metadata, and taxonomy assignments if you have it. This part is optional. Phyloseq can also take a tree file if you want to. Generating a tree file isn't really part of DADA2, so I've left it out here. But if you Google the Ben Callahan in Faculty of 1000 several years ago published a paper that's just sort of an entire workflow from raw sequence analysis, from raw sequence data all the way through to many different kinds of ecological statistical analyses using Phyloseq, so including tree generation. So I'll encourage you to look for that if you want to create trees. And then just some basic, if you go through and create this, the assigned taxonomy takes a little bit of time, so I'm going to skip this, but if you create this, then this is just an example of an alpha diversity that you could plot. So, okay. So that is DADA2. Let's close this for now, or minimize that for now, and get back to the presentation. So we did DADA2 and primarily DADA2 and R. That was the important part, is our analysis. So QIIME2 is it's a big thing. It's less of it's less of a software, less of a pipeline, and more of a community of options. So even more than the original QIIME, QIIME2 has really expanded by adding the ability for people to write plugins, and since they implemented the plugin functionality, they've as it's called, dog fooded it. So all of the core functions are implemented as technically plugins, but there's a lot of stuff, and more stuff is being added, and so they're really leaned in hard to the community aspect And so you can find a lot of tools similar to R. You can find a lot of tools where people have written tools for other things, so there are a lot of R now tools that are available only in R normally, where people have written plugins for QIIME2 um, that you can find. So um, some core concepts before we start into QIIME2. If you're familiar with QIIME1, or even what we went through with DADA2, where everything is operating on individual files, QIIME2 really, because that was how QIIME1 operated, was basically a loose collection of Python scripts, Python programs, and you just had to sort of know your way around things and make sure you were doing everything correctly. QIIME2 tries to help people with that, one, by uh, uh, adding type information to analysis products, and two, adding metadata. So instead of operating on standard data files directly, so for example, FASTA files or um, uh, those sorts of things, the very first thing you do in QIIME is import your data, or QIIME2. The very first thing you do is import your data into an artifact. And so their artifact files are called QZA. So um, I don't actually know, they don't actually explicitly say this anywhere that I've seen, but the file extension um, uh, logically is QIIME, Q, Z for zip, and A for artifact. So it's a QIIME zip artifact. So the zip part is important because the QZA files and the QZV files that we'll see in a second here are literally just zip files. So you can rename it, any of the QZA or QZV files, to .zip and use a standard unzipping tool to open it, and there's a folder inside that has a whole bunch of stuff. And we'll see that in a little bit. But the QZA files contain the actual data files as well as metadata. So when you import, and we'll see some of this later, but each artifact file is associated with a type, which prevents it being input. So if you try to input the wrong type of file into a certain analysis step, it'll yell at you. But it'll yell at you in a useful way and saying, hey, you gave me this type of file when I really need this type of file. And the last thing here is provenance, but it's really about reproducibility. So each QZA file maintains a list of all the tools that were run before it, so you know exactly how that file is generated. And you can look at any of that with the website view.qiime2.org is their sort of standard tool. It's entirely done in the browser, so it's not like it's giving data back to them. It's all done in JavaScript in the browser, so all the computations and data stay on your computer when you go to that website. But that's their standard tool for viewing both the artifact files, the information stored in the artifact files, as well as the visualization files. So, yeah, okay. Okay, so as I said, the semantic types that I mentioned earlier prevent incorrect analyses from being performed. So if you have a feature table type that requires with presence absence data, so ones and zeros, it won't work for an analysis that requires counts, so 1 through n kinds of things. So this is an example, so you can see here feature table, capital F, capital T, in brackets, presence absence. So sort of like major type and subtype. And you'll see this is the general format for these semantic types, and you'll see this throughout. Every analysis step, when it finishes running, will print out the output file and the semantic type of that output file. So methods generate artifacts, and artifacts can be input into other methods. And you can chain that as far as it'll go. Visualizers only generate visualizations, and that's the end. So you can't put a visualization into anything else, into any other tool. you can only visualize it. So I already mentioned about the plugin architecture. So this is sort of just a conceptual overview. And again, this goes back to the flowchart I showed earlier. This is just more specific to QIIME2. So again, we start with raw sequences. We demultiplex if necessary. So depending on the kind of sequence data, this isn't so much of an issue anymore with the MySeq or HiSeq machines, but the old 454 machines used to multiplex all the sample sequences into a single file with prepended barcodes that you would then have to go and bioinformatically pull apart based on those barcodes. So they have a number of different ways to input sequence data. After you've and quality filtering is in here too. So once you've got your sequence data, if you have paired ends, join your pairs, do quality filtering, do denoising or clustering. So like I said, they implement an interface to DADA2. They implement their own deblur, and honestly, again, even though this is literally their tool, deblur is QIIME2. The authors of QIIME2, it's their tool. If you go and look at some of the tutorials, they mention that if your data has this particular feature or this kind of thing you see in your data, then the QIIME2 plugin for deblur isn't enough. You'll have to go and actually run deblur yourself if you want to do certain operations or edit certain parameters. So interfaces are almost always limited, because if you're going to implement a 100% faithful reproduction of the thing you're interfacing with, why wouldn't you just use that? So that's somewhat facetious, but in general, interfaces are usually have some sort of limits. Okay. Yeah, things always take longer than we expect. So we'll try to run through these here. But you can see the general idea, the overall, the overall flow for the analysis. So we will go ahead and do this. So again, if we go to the GitHub page, we can go to the QIIME2 folder. The metadata file is important. It's just it's a TSV file that lists the sample IDs and various metadata about them. So we can talk about it here, but yeah, so sample ID, gender, day, and then a categorical mapping of day to, since some of the tools won't take numbers, they don't like columns that have numbers, so we've kind of mapped to early and late, so you get a categorical variable in when there that you can use in certain analyses. But that's important for some of the steps, so you'll need to download that as well. Okay, and then the QIIME2 process here is just sort of the file I will be going through. I'll paste it here. It's a shortcut. Okay. Yeah, so I'll be going through that. So we will then switch over to the console to start running things. And let me pull up my own copy of this. Okay. All right. So the data set is the same here, so you can use the same data. I've just duplicated it. So this is, I've already done everything here. And the deblur step takes 5 to 6 minutes, so I'm not going to make you guys sit here and watch that. But you'll notice here, so this data folder is the same data folder, exactly the same data folder we used for the DADA2. I just duplicated it and copied it inside of the QIIME2 so its paths are easier. But it's the same data set that we just used. Okay. So as I said, the first step is gaining entrance into the QIIME2 ecosystem by using the QIIME tools import. And they actually have an entire page, an entire tutorial page just on the importing function. So so QIIME tools import is the general tool, and then you give it a type. So in this case, we're importing paired end sequence data with quality. So this is FASTQ files instead of FASTA files. The input path is the data folder that I just showed you. The input format is Cassava 1.8, so that's the Illumina sequencing format. And then we're okay. And the output is the paired end. Yes, sorry, I knew that was a mistake. I was thinking I was trying earlier, and I realized I didn't need to do that. We will remove the metadata as an input file there since it's unnecessary. And so just we give it the semantic type, the path to the data files, the format for those files, and then the output path. So it's the files are all separate, so they're technically pre-demultiplexed, which is where the demux comes from. I've just added paired end. Not really necessary since we don't have a single end to compare it to, but just to be consistent throughout. So we can generate a visual representation of the metadata file. So you'll see here, so QIIME metadata tabulate, and we'll see this function over and over again. It's useful for a number of different steps. So we're inputting, the input file is the metadata table, and the output file, the output visualization file is a QZV metadata file. So we can generate that, and then we will bring up the view.qiime2.org. Okay, and take the file we just generated, QIIME2. Let me just open this specifically. There we go. Okay. Oh, right. Okay. There we go. So we can just focus on that folder. So the file we just generated was this tabulated sample metadata.qzv. We can look for that tabulated sample metadata.qzv. So we can literally just take this and drag and drop it into there. And there's our, we could just as easily open the TSV file in Excel or LibreOffice or something. But this is another way to visualize the metadata file. And also the other nice thing is that you can pass these, you can share any of these artifact files with other people, and they can go and plop them into QIIME2 view, you know, and they can download the metadata, the original TSV file, or you know, they can sort each of the columns. So it's a nice interface. So we'll just go back to the view folder or the view website and leave that up there. So the next step is to visualize equality information, which again we did with already did with DADA2, but we can see how that what different options are available. So you'll notice here we're again the -o visualization. So we're generating a file called demux paired end.qzv. So we'll go look for that. Demux paired end.qzv. And so you'll notice there's a an accompanying artifact file for the visualization file. So we can drag that there. So it's just the number of reads, the sequence count summaries for the forward and reverse reads. These should be identical. If they're not, you have a problem. And we can see again here that we saw with DADA2 that 2 of these samples are just, there's something wrong with them. So, and then you can also see interactive quality plot. Again, same general idea that we saw with DADA2. It's a little bit more interactive. You can see the quality scores changing in the table below as you move along, but in general, again, average quality is pretty good up until around base 240, and then it starts to dip down below 20. And the reverse reads, again, around base 160, the average quality starts to drop a whole bunch. So it's just nice to see the same thing from 2 different tools. So we will go back to the console. Okay, so we took a look at the quality information, and so now the next step, as we saw in the formatting, is to join paired ends. So we will do that now. Okay. Yeah, sorry, I'm glancing back and forth between the comments. So the question was asked, does this console need to be installed on a PC or Mac? So yes, this is this comes the console or the terminal is an integral part of Mac OS and Linux or any of the Unix things. Windows has a version of this. In the past, it was very limited and unpleasant to use. Someone mentioned earlier they now have the Windows users from Windows 10 on have access to a Linux subsystem. I haven't played with it for a long time, so I don't know how much better it's gotten. So there is sort of a version that you might be able to use in Windows of a terminal like this, but otherwise it's probably better to get a Linux virtual machine and run it inside of Windows. Then you're guaranteed to be able to run everything as you would expect. Okay. All right, so yes, join paired ends. So they use the great tool vSearch, which is a very flexible tool for doing a lot of different sequence alignment type things. You can align sequences, you can search sequences, you can join paired ends. vSearch is more of a toolbox than than just a single tool. So we'll run this. So again, so we're running QIIME vSearch, the join pairs method in vSearch. We're importing we're giving it the raw sequence data that we imported earlier, and we're outputting the joined sequences to a file called demuxjoin.qza. So we'll run that. That will take a second. Yes, I can increase the size of the terminal. I can increase the size. Let me see if I can increase the font size as well. Thank you for bringing that up. Is that any better? Okay, yeah. Okay, let me know if I need to increase it some more or if that's okay. All right, so again, and I haven't mentioned it, but you know the green here you can see is it's listing the output file and the semantic type of each of those output files. So we know we have now a demuxjoin.qza, and then we can generate a summarization of that. Now I did notice while I was doing this, so it is supposed to. So we can take a look at this. It is supposed to generate a quality graph similar to what we saw with the paired ends. So we're looking for demuxjoin.qzv. So we'll drop that in there. So this is just again, similar to what we saw before. So it's instead of forward and reverse, it's just forward, even this really paired or joined. So we can just see how many reads are left over after the paired joining. So this is basically, again, similar to the table we saw in DADA2. It's not quite as convenient having everything as we had everything in one kind of big table, but you can kind of keep track of, make sure you don't lose a lot of stuff as you join. Again, these two are just terrible, so they are what we expect. Now this is supposed to have an interactive quality plot. I'm not sure why this looks terrible like this. It should look more or less similar to what we saw before. I don't know if it's some bug or something, but yeah, this just wasn't working for me. So maybe it'll work for you. Maybe it'll be a bug maybe it's a bug that'll be fixed later. So, but again, you can always run fastqc on the raw data or on the joined reads if you wanted to check that out later. And actually, let's see. Yes. So I just made a copy earlier of the QZA, which is the artifact file. I renamed it to demuxjoin.zip and unzipped it. And so this is this joined folder is really what's inside of the demuxjoin, the output from the vsearch joining step. So it's got checksums, which we talked about last week. So all of the input files, it has MD5 checksums, so you know that you can always go back and check and make sure there's no data corruption. There's a data folder, so all of the top level stuff is the same for any artifact QZA file that you open. The checksum is going to have the data folder. It's going to have a metadata YAML file. It's going to have a provenance folder. And then this will be different. And it actually does, sorry, so it actually does, so this QC folder, I actually ran myself. I ran fastqc on it to take a look at the quality data of the, so I ran fastqc on each of the individual samples and then multi-QC to merge them all together. So we can take a look at that. So this is what I was expecting to see in QIIME, but for some reason it wasn't showing up. So we can see here, this is the result of that quality dip in the end of the reverse reads. But this is generally what we would expect. And overall, even with that dip, the average quality for the sequences is still pretty good. So, okay. So that is joining the paired ends. Okay. So quality filtering we will do on the joined sequences. So here we're filtering on the Q score that comes with the FASTQ files. We're giving it the joined FASTQ files, and we're outputting two files. We're outputting the filtered version of the joined files, and we're also outputting a stats file that just summarizes the information from that. So we can run the quality filtering. Okay. All right. Let that run for a little bit. Now this next step, we already obviously ran DADA2 in its native environment. So instead of using, so we'll, for QIIME, for the QIIME step, we will run generate ASVs with dblur. As I said earlier, even for this reduced data set, it takes a while. Since I've already generated the data, we don't need to sit here. But this is the command we would use to generate that. But we won't run that. We will simply use, I just hit Control-C there to exit out of that particular line. So we can already see here, so we have the representative sequences and the, which is the ASVs, like we saw with DADA2, and the table here is just the sequence table, the sequence counts table again, that we generated with DADA2. And so this is the nice thing, is that, and that's why I had that code at the end of the processing pipeline for DADA2 to export the representative sequences and to export the counts table, because then you can run the whole DADA2 pipeline up until generating the ASVs, the Chimera depleted ASVs, export the sequence table, the sequences, and the count table, and then pick up in QIIME 2 directly after this step. Okay. So then we will visualize the read counts, and it's similar to what we did with DADA2, so we can see what happened with deblurring. So first we will tabulate the information from the joined and filtered data, and then we will do the same thing for the results from deblur, and so we'll see what happened between each of those steps. So we can load the demux joined filter stats.qzv into here. So we can find, okay. So the demux joined filtered stats.qzv, yeah, matching there. So we can load that into the QIIME viewer. Okay, so yeah, so the total retained reads is mainly what we're looking at here. So again, as with DADA2, we didn't lose too many from each sample, which is good after filtering. And then after deblurring, oh, and then yeah, so this is what you would see once you output, or if you actually ran deblur. This is the output that you would see. So okay, all right. So the other file that we generated was deblur stats.qzv, so we can visualize that now. Deblur stats.qzv. Okay, drag and drop that there. Yeah, okay. So the main thing, so the nice thing about this is you can mouse over any of the column headers and see what each of these, what each of them mean. So the unique reads deblur is basically the ASVs. It's the same as the ASVs in DADA2. So these are the unique reads that are found. So these are the ASVs, and then this is the number of reads associated with each of those. So the 152 unique sequences are associated with 3,000 actual reads for that sample. And you can see how many were lost to chimeras, etc. And they also do a similar, we'll see this with the Mothur data set, but they do a broad match against a broad sequence database to make sure things are actually bacteria archaea, and we're not matching sequences to eukaryotes, for example. So there's a number of things there to explore in that the deblur output. Okay, so go back. Okay, so we can go back to the console. Okay, so someone's saying you're demultiplexing your data with vSearch. So no, we're not demultiplexing the data with vSearch. It's kind of a yeah, the file naming may not have helped there. So I just called it demux or demultiplex because it was pre-demultiplex. So you're right, Illumina does this for you. We're using vSearch to join the paired ends, not demultiplex. So when you see a file that's called demux-join-filtered, it means that the data stored inside that artifact has been demultiplexed, in this case pre-demultiplexed by Illumina, and then joined by vSearch, so the paired ends are joined, so this is no longer single end data, whereas before the demux paired end file contained the forward and reverse reads. So the demux joined, and then finally this artifact contains joined data that was also then filtered. Okay. But yeah, so this goes back to the issue of naming files that I mentioned. So that file naming system made sense to me. It may not make sense to someone else. So if you're working with multiple people, agree on something that you can all be happy with in terms of file naming. Okay, so the next step after that is to summarize the ASV results. So we can generate So this takes in the feature table, so the counts, the counts of ASVs per sample, and we add in the metadata file. This is the metadata file that I had you all download earlier. And so it merges the metadata file with the count data, so you can take a look at that. So we look at table.qzv. Wrong drag and drop location. There we go. Okay. So this is the visualization for that. So again, we have 23 samples left over. So out of the original 24, as we saw earlier, after filtering and Chimera checking everything, one of the samples had no reads left. The number of features here, features means ASVs, OTUs, whatever. So we have 251 de-blur ASVs. And total frequency is the total number of reads, or the total count of reads, 121,000. All right. And just some additional graphs and histograms. And we can look at this all by sample ID. And this particular, this one is important. This gets back to the normalization question that someone asked earlier. For the alpha and beta diversity, QIIME2 has a workflow script. QIIME2, they typically like to, for normalization, they typically like to rarefy. So rarefaction is pulling sequences from samples randomly over multiple rounds to achieve a particular consistent read depth across all the samples. So that way, that's one method of normalization for different numbers of read depths. And you can see here, so female 5 day 0 had 12,000 reads, whereas well, we'll ignore this guy here, but female 8 day 149 had 1,300 reads. So if you're going to do rarefaction, you need to make some decisions about how many samples you're willing to lose, because if you set your rarefaction depth at 2,200, then this one will drop off. So if you're willing to lose a few, if you've got some, so like this one, for example, there's no way we're going to rarefy to 18, because that would be ridiculous. So we will rarefy to 1,300, but you have to kind of you have to weigh losing samples versus including more sequences in your rarefaction. So that's what this table is useful for. Okay, so we will do that now. Or no, I'm sorry. Yeah, so we can do we don't need to do this, but I'll just put it out there. It's in the process file. But if you want to look at so we were just looking at the feature counts table. If you want to visualize your representative sequences, your ASVs, you can use this command as well. But we will skip that for now. So phylogeny is built into QIIME2, so generating a phylogenetic tree. You can run that, so it does uses MAFT. The MAFT uses MAFT for multiple sequence alignment and uses FastTree for actually generating. So it first takes your representative sequences or your ASVs, aligns them to themselves using MAFT, and then uses FastTree to generate an unrooted tree, which is this you see here. So these are all output files. So you get your masked alignment. So you get your alignment file, your masked alignment file, your unrooted tree, and then finally a rooted tree. Since most tools that use a tree for analysis want a tree with a root. So typically for unrooted trees, the heuristic for generating a rooted tree is just to take the midpoint of the highest level. That midpoint doesn't necessarily mean anything. It's just a way to generate a root. That's typically how a rooted tree is generated from that. So someone is saying, can you use these same pipelines for nanopore sequencing data, or minion data? You know what? I will leave that to next week for Olivier. He's a lot more familiar with that than I am. So it is possible, but it's not necessarily easy. I believe I have to go look. I'm not sure if actually I'm not sure if DADA2 can handle uh, nanopore data or not. I have read some people trying to do it with QIIME2, but I'm not 100% sure. And ultimately, it's sequence data. So the real issue is is not doing something with the sequences. It's dealing with the kind of data that's generated. So all the filtering and that kind of stuff is vastly different than the quality data that's generated. So if you can do some pre-processing, okay, yeah, so Olivier says he hasn't used those pipelines with Nanopore, but he said he can add something to that to next week. So that's good. We'll leave that to him, so we can keep going here. Okay. All right. So we will skip that for now, the tree generation, but that's how you would generate it. And then, like I said, QIIME has a workflow script to generate a whole bunch of different metrics, alpha and beta diversity, which we'll discuss in a little bit here. This sampling depth parameters, the rarefaction depth that I mentioned. So all the samples following this step, all the analyses that use this data, all the samples will be limited to 1,301 sequences to match the sample with the lowest sampling depth. So all the analyses that are produced from this will be based on that number of sequences per sample. This does take a little bit of time, so I won't generate that now, but I already have that data there. So we can go through and just real quick, we can take the one of the alpha diversity metrics, the face phylogenetic diversity. We can generate that here. And then you can see that it outputs to the core metrics results folder the faith PD group significance.qzv, so we can take that. Right, it's under biometric results, faith, PD group significance. So this just takes the alpha diversity metric, faith's phylogenetic diversity, and lets you visualize it, lets you group it by metadata columns. So there's clearly no difference in gender between the male and female mice in terms of phylogenetic diversity, alpha diversity. And we can see there's they run Kruskal-Wallis, the non-parametric version of ANOVA. So for gender, there's no difference. But if you look at when, so early versus late, then that does appear to be significant. That the based on the faith's phylogenetic diversity, the earlier samples have higher diversity than the later samples. Okay, so there's a bunch more in there that we can do. We're not going to finish it. Assigning taxonomy, again, is a whole separate thing, and you can download their classifier. They recommend generating your own classifier, especially since green genes is dead basically now. It hasn't been maintained since 2013. So I think it's pretty much officially dead. So they recommend using Silva. One of the things I did forget to mention, since we didn't do the taxonomy generation for DADA2, is that Silva is generally sort of the most comprehensive marker gene database. So a lot of people use that. One of our later presentations will be about the genome taxonomy database that has really sort of revolutionized, not to say revolutionized, but they've taken a completely new look at taxonomy. And so one of the people behind the GTDB will be talking later on. So we will have, I'll show that here. But I bring that up to say that, yes, so this is week 3, 4, 5, 6. So week 7, we'll be talking about the genome-based taxonomy and the genome taxonomy database. So DADA2, Ben Callahan has put together a taxonomy of Silva using GTDB. So, and I included both in the resources. So you can download the original Silva training data set and taxonomy, or you can use the GTDB taxonomy there. So, okay. So the last thing that we need to go over is Mothur. We're definitely running over time here. So again, this is, we may actually just skip this. The whole process is, like I said, available. This is the general idea. So data preparation, the first step is to go take your data and they join the paired ends at the very beginning. So the merge samples. They do their standard operating procedure. They filter on sequence length, base quality, homopolymers. One of the unique things about Mothur is that, well, less unique now. Actually, QIIME 2 does the same thing now. QIIME 1 didn't used to, but they take the database file of aligned sequences, aligned reference sequences, and trim them to the region of interest. So if you're looking at the V4 region, they take the full-length 16S sequences and trim the database sequences themselves to exclude anything outside of that region that you're looking at. And then they go through and align. So they do some additional quality control. They look for chimeras. They also Mothur also uses the vSearch chimera checking, although they implement the algorithm themselves in C++. And they also run again a broad check, kind of like dblur does, 80% similarity to make sure that your sequences are not matching against eukaryotes or mitochondrial DNA or something like that. They also go through, this is something that again this actually is somewhat unique to Mothur, is that they Pat Schloss recommends including mock sequences, or a mock community. So creating a library of known organisms. So he has his mock community is, I believe, 20 organisms of approximately known abundance. And so they actually go through, and for every 95 sequences, they recommend including one of these mock community samples. So in the data set, you can download, there's one mock community, but they actually use that mock community to assess, since they know what organisms are there and about how abundant they are. They can use that mock community, the sequencing, the sequencing result compared to what's expected, to assess error rate for the overall sequencing process, and then use that in their clustering process. Now, as I mentioned earlier, Pat Schloss is somewhat reluctant, has some issues with the whole ASV thing, so he's preferred to stick with OTU clustering. Oh, am I muted somehow? No, I'm not muted. Are other people still hearing me? Someone said they can't hear me. Okay. All right. Other people can hear me, so sorry. I don't know what happened there. But it looks like most people can hear me, so thank you. Okay. Pat Schloss, Dr. Schloss has decided to stick with, instead of the ASVs, a more of the clustering approach based on sequence similarity. Although, like I said, he has moved away from the original clustering, or not moved away, but kept up with the times, and created a new, there's a 2017 paper he has on a technique called OptiClust, that uses a more advanced clustering method, along with the Matthews correlation coefficient, to, in an iterative process, generate more reliable OTU clusters than previous processes like uClust. Mothur has a slightly different, like I said, they don't do the ASV thing, although I will say they do implement a version of ASV clustering. You can do that if you want, as opposed to the OptiClust method. So you can choose either one, and then again, you can classify your sequences to a database. He recommends Silva, and one of the other nice things about Mothur is that, as I said earlier, one, they implement everything on their own. So everything, as much as possible, they re-implement for Mothur. But two, it's definitely worth exploring. They have a number of alpha and beta diversity and statistical methods that are hard to find in other places. So it's worth taking a look at some of the methods they provide in Mothur, some of the unique methods for analysis that they provide in Mothur that are hard to find in other places. So apologies, time is kind of running out. I do want to get to the ecological statistics. We'll skip going through interactively the Mothur pipeline, but you have the whole thing there, so you can take a look at that and run through it yourself with the data that I put together. So again, same data that we used before. So we will finish that and switch over to the ecological statistics. So this is the ecological statistics. Let's get this started here. Okay. Okay. And again, we'll try to be fast on this. But this here is, and again, this is kind of echoing what I said at the beginning about methods and their, 1,001 tools. That applies to statistics as well. So what we're really trying to do here, okay, yeah, so Isabella. So personally, I use, what I've typically used, I started out in this world using QIIME. Not by choice, that was just the lab I was in. They were already using QIIME. So a lot of the QIIME versus Mothur stuff, it's a Pepsi versus Coke. But I am much more familiar with the QIIME side of things. So I typically in my own analysis will use DADA2 and R to generate ASVs and the count tables, and then I will export the ASVs for taxonomy assignment in QIIME, and a lot of the downstream. So for the downstream statistical analyses, I'll use a mix of QIIME and R. So Phyloseq and vegan, those sorts of things, and any of the other R libraries. So it's useful, like I said, to have things like biome tables or just count tables, because they can be imported into pretty much any tool at that point. But that's the general sort of approach that I typically use for most things. Okay, so yeah, so the ecological statistics, this is not meant to be a talk about the latest and greatest. This is meant to, again, as I said in the previous talk and the previous set of slides, give people a grounding in the techniques, the general techniques people use. If you don't know this stuff, then, or rather, if you do know this stuff, then it'll be a lot easier to read papers and understand what tools people are using. And then once you know this, you can go further into more of the new things, more of the new techniques, the new tools. But this is where we need to start. So there are quantitative and qualitative measures. Quantitative measures use the abundance, the counts the count table that we generated. Those are quantitative measures, and qualitative measures use presence absence. They both have pros and cons, and really the cons are the only cons are you're using it for something that it shouldn't be used. So so like there's a presence absence. We'll get to the Jacquard metric. Is very useful. And quantitative measures, they tell you different things. Yeah. So and I've given some examples there of when to use each. And this is the other thing too, is that in all of these tools, there's no one tool, one ecological statistic, one metric that will tell you everything, right? So this really is about exploratory data analysis. Is go through each of these tools: alpha diversity, beta diversity, differential abundance analysis, networks, all these things that we're going to talk about. They all tell you something different, and so you can use all of those tools, generate data using all of these statistical tools, all these ecological analysis tools, to build up a picture of what you're looking at. All this data is very high dimensional, and so all these tools are really just giving us one view of what's going on, not just from a dimensionality aspect, but from the aspect of understanding different parts of what makes these microbiomes tick. And so there's no the main takeaway here is there's no one tool. Use a lot of different tools and use them correctly and understand what they're telling you, and use them all together to build a story about how your environment, your microbiome, is acting in its environment. So we've seen this before, alpha diversity. This is richness, species richness, you'll hear a lot. And so microbial ecology, alpha diversity, is the diversity within a single sample. So it's when you calculate alpha diversity, you're calculating it for a single sample. So what is it? Richness is the number of species in a community. So see here, community 1 on the left, just visually has a whole bunch of different trees. You can see four different trees, A, B, C, and D, whereas community 2 is much less rich. It only has 2 trees, right? But not only that, as you can see in community 2, of the 2 types of trees, one of those types of trees is very dominant, which you don't see in community 1. So community 1 is richer because it has more different types of trees. But as we'll see, community 1 is also more even than community 2. So going back here real quick, so calculating richness, there are a number of different richness estimators. There's the observed species, which is just the number of non-zero entries in the count table. There are richness estimators, so Chow1 is an estimator because and a lot of these tools for alpha and beta diversity we'll see later were really developed for classical macroecology as opposed to micro or microbial ecology. But a lot of these tools still apply and can still be useful, provide useful descriptions for the communities that we're interested in. And a lot of the same problems occur, right? So if you're going out and counting, as I said earlier, trees or bears and tigers and rabbits, it's physically impossible in an open environment to know whether or not you, and likely not, have accounted for all of the organisms in a particular area. So they're going to be observed and unobserved. And of the things that you observe, generally, if there's more of them, you will likely count them more often. But we know that some were observed and some were unobserved, and so we have to do something with singletons and doubletons, or single count ASVs, for example, which is why the pooling was important. You get more observations pooled across multiple samples. But this is all just to say that a lot of the considerations that were used in macroecology are also applicable to microecology. So again, evenness, and there are different. So there are different metrics for richness separately as well as evenness separately. But alpha diversity combines both richness and evenness into a single metric. And so Shannon's index here takes, so P is the relative abundance, moderated by the natural log of relative abundance, and summed over all of the organisms or ASVs or OTUs in each sample. And so you get a single number, H naught, for each sample. The problem with a lot of these is going from the mathematical descriptions to some sort of intuition. So the interpretation for Shannon's is the uncertainty involved in predicting the species of an individual sample at random. Simpson's index is the second alpha diversity index that can be interpreted as the probability that 2 species or OTUs taken at random from a data set of interest represent the same type. And so if you have much higher diversity, then that probability will be lower. If you have more, if you have 100 species, then your probability of picking 2 that are the same type is much lower. If you have many fewer species, then the probability that you pick 2 that are the same type is going to be much higher. So we generated this earlier with QIIME2, face phylogenetic diversity. It's an alpha diversity metric that uses the phylogenetic tree. And so it's calculated by the sum of a branch length covered by a sample. So you take the branch lengths, and these are sort of the branch lengths are generally related to phylogenetic distance, evolutionary distance between the things below and above. And so you can go through. If a sample has feature 1, 3, and 4, then you just start from the root and add up all of the branch lengths, and then compare those. And so we see here we have 2 samples that have different numbers of each of the features. So we add up all the branch lengths, and we get 2 different alpha diversity scores based on phylogeny. So this, just in general, phylogenetic measures are often more useful because they do take into account especially with microbial ecology, the complex relationships, and we'll see in a second here with Unifrac. But if you have for some of these metrics, if you're just looking at the number of species, the metrics aren't necessarily taking into account the relatedness and the fact that if you have two species of the same genus, they're probably there because they're able to survive in that environment better. There's some nutrient source or something that enables them to survive. And so you're going to have, if you see one species of a certain type, you're more likely to have another species of that same genus, just because they share so many genes, so many functional capabilities, and so we'll often share the same ecological niche adaptation and be able to eat the same kinds of foods and that sort of thing. So in general, it's often better to use phylogenetic metrics. So as I said, since alpha diversity just generates a single number per sample, we can do standard single variate statistics and descriptors. So just here we have and I'm completely blanking here, box plots. So we can see right off the bat that, and this is for a different data set. This is for the moving pictures data set of humans that compares gut to hand to tongue samples. And so we can see right away that the gut samples have much lower diversity than the hand samples. And the tongue is sort of in between the gut and the palm samples based on face alpha diversity. And since we have univariate statistics, or since we have a single number, we can use standard univariate statistics. So typically you'll see people use non-parametric statistics here, simply because the nature of this kind of data does not lend itself to normal distribution. And so typically you're forced to use non-parametric statistics unless you can transform the data into some sort of normal range. So Kruskal-Wallis, as I said earlier, is sort of the non-parametric version of ANOVA. And so here we can compare and get that, there's the group-wise, there's a difference. And this is in QIIME2. And so they go through and do group-wise comparisons, and then they do pairwise post-hoc comparisons as well. So again, as I said earlier, there's no one tool for analysis that will tell you everything about the organisms and the environment and the ecology that you're looking at. So there's all kinds of different patterns of interest and different tools that can tell us about those patterns of interest. So what drives the patterns of interest? And we'll go through each of these three, but there's exploratory methods, interpretive methods, and discriminatory methods. So the exploratory methods explore relationships among the organisms in the environment that we're interested in based on the values of variables measured in those objects. So for example, this is where we get into beta diversity, which we saw a little bit earlier when we were talking about QIIME. So whereas alpha diversity is a within sample metric measure, beta diversity, or also known as community dissimilarity, is a distance measure between separate samples. So community 1 and community 2 here are like a gut sample versus a hand sample, for example. In this case, just simplified to use trees because it's easier to visualize. So we've got multiple different communities here. And notice there's some numbering differences here. We've got 6 different communities here. And the arrows here represent the total set of pairwise differences between all of the samples. So this is the important part about beta diversity or community dissimilarity, is that whatever metric you use is you apply it pairwise between all the samples. And so when you do this, you get what's called a dissimilarity matrix. So if you look at this down, you have a matrix of samples. So the columns are the same as the rows, and the diagonals are all 0 because those are all the self-dissimilarities. So all the samples are equivalent to themselves. But then the lower half of this matrix, the lower triangle of this matrix, is all the pairwise dissimilarities between all of the samples separately. And so this dissimilarity matrix is used. So the other thing here is that whereas alpha diversity is a univariate metric because you have one number per sample, here with beta diversity you have multiple numbers per sample. You have one number for every comparison that was made to all the other samples. So this is now into the realm of multivariate statistics, which is a very different set of analysis methods. So again, these are the first 3 here, Jacquard, Sorensen, Bray-Curtis, are very old metrics that are developed for macroecology, but they can still be useful even in microecology. The fourth there, Unifrac, is a newer method developed by the group behind QIIME. Catherine Luzapone is the first author there behind the original Unifrac. That was specifically designed for microbial ecology, beta diversity metric for microbial ecology. One thing to note here is that the use of the word metric it needs to be noticed. So Jacquard follows the triangle inequality. You can look it up, but the definition of a metric is that it follows the triangle inequality. So some of these do and some of them don't. And so Bray-Curtis is not technically a metric. It is a measure. Unifrac does follow the triangle inequality, and so it is a metric. So Jacquard, just going to go through some of these real quick. Jacquard is a measure of shared species, but only at a qualitative level. It only measures presence-absence. Bray-Curtis is the same idea, a similar idea, where it looks at shared species, but it is a quantitative measure. So instead of just looking at presence-absence, it is actually looking at the number of shared species in addition to the fact that they're shared. Here, a 0 for Bray-Curtis. So the original measure here that's listed here is a measure of similarity. So if you want a dissimilarity for use in beta diversity, then you have to subtract 1. So 1 minus the BC metric there. So the Bray-Curtis dissimilarity is always in between 0 and 1, where 0 indicates that two sites have 0 dissimilarity. In other words, they share the exact same number of each type of species. And at most, 1, a dissimilarity measure of 1, indicates that the two samples have complete dissimilarity. They share none of the same type of any of the species. So they're completely dissimilar. Unifrac that I mentioned was designed specifically for microbial ecology, and it is a phylogenetic measure. And there, it comes in 2 flavors, weighted and unweighted. Unweighted simply is very similar to the Faith's phylogenetic diversity that we looked at earlier. It's the sum of the unique branch lengths over the sum of the observed branch lengths. Another way to say that is the fraction of branch lengths that leads to the descendants from each single community, but not from both communities. So it discounts the branches that are shared and focuses on the branches that are different between the two communities. And so you can see here on the left, completely similar communities have a unifrac measure of 0 because the blue and red communities share the exact same branch lengths. All the OTUs are shared between them. And then at the far extreme, the two communities, red and blue, are have completely different branches or occupy completely different branches from the root to the tips of the trees. And so they have a maximum dissimilarity of 1 in unifrac. Now, like I said, there is a weighted version of this that weights the branch, that includes the branch lengths and weights the abundance differences of the branch lengths. I'm sorry, weights each branch length by the abundance differences of the branch along the tree of the communities. Now, unifrac, as you can see here, the citation was created back in 2005. There have been a number of modifications to this that you can look up. So one example, there's a modification of unifrac that looks to test if the two communities are different, but more specifically, if the sequences are randomly distributed among the leaves of the tree. So again, there's a number of different modifications, but like I said earlier, understand what unifrac is first, and then you can go and look at all the different modifications. This applies, again, all the way throughout. So what do we do with the diversity, the beta diversity measures? One of the main things we can do with them is ordination or visualization. So we take our multi-dimensional metric, and if we have 100 OTUs, then we have a 100-dimensional matrix. And so we have to have some way to collapse that down into 2 or 3 dimensions so we can physically visualize it. So there are a number of different ordination methods, but they all take some sort of statistical approach that attempts to order objects based on the values of the variables measured in those objects. These are also often called gradient analyses because these algorithms look for the main gradients of variation in the multidimensional data space and then arrange the objects in a new system of coordinates with each gradient serving as an axis. And typically, the first 2, 3, or 4 of these gradients account for the majority of the variance in the data. And so we'll typically see that in nearly all cases, the majority of the variance in the data sets can be visualized in 2 or 3 axes. And so this is one of the main parts of ordination. One of the main draws of ordination is that it's dimensionality reduction coupled with a straightforward visual interpretation of the objects. So samples that are closer together are more similar. That's the general idea behind these ordinations. Okay. Yeah. Okay. So principal coordinates analysis is one of the main, also called metric dimensionality MDS metric. I'm completely blanking on that now. But yeah, principal coordinates analysis is one of the most commonly used versions of one of the most commonly used ordination methods for dimensional error reduction for ecological statistics. So this in particular is a representation of the principal coordinates analysis of a Bray-Curtis dissimilarity matrix. So again, objects that are closer together have smaller dissimilarity values than those that are placed further apart on the plots. So not only that, but these ordination methods attempt to preserve the distance generated from the higher dimensional matrices. So PCOA produces a set of uncorrelated or orthogonal axes that summarize the variability in the data set. So each axis has an eigenvalue whose magnitude indicates the amount of variation captured in that axis. Now this is getting into matrix math and that kind of thing. So don't worry if you don't understand that. You can go read it later, read about it later. But the proportion of a given eigenvalue to the sum of all the eigenvalues is how important each of the axes is, and this is where we get. You'll see with these principal coordinates analyses that each you can see here. So axis 1 is representing 24.8% of the variation, whereas axis 2 is representing about half of that. And this is a plot produced by Phyloseq, and so they actually scale the axes to indicate their variation as well. But yeah, so unfortunately with PCOA, it changes things in a way that we can't get information on the original variables after transforming to the new axes. So that was one downside. That's it. MDS, multidimensional scaling, metric multidimensional scaling. So metric multidimensional scaling is another name for PCOA, or PCOA is a version of MDS. So non-metric multidimensional scaling uses ranks instead of the original dissimilarity values. So NMDS attempts to represent as closely as possible the pairwise dissimilarity between objects in a low-dimensional space, instead of maximizing variance. So it's an iterative process that starts in the 100 dimensions and goes down to the 99th dimension and says, okay, well, how can I group these points so that the distances are as similar as possible to the previous dimension? And it does this iteratively as you go down. And so you'll see there the stress value. So the stress value indicates more or less how faithfully the distances between the objects are preserved in the lower dimension versus the original full set of dimensions. And so stress values less than about 0.15 are generally considered acceptable. So why would you use PCOA versus NMDS, since they're basically doing the same thing? In general, visualization methods are visualization methods. Use the ones that represent your data. But one advantage that NMDS has over PCOA is that often in data sets with many overlapping gradients, NMDS can often produce a clearer overview of the data distribution. No, QIIME2, I do not believe QIIME2 has a way to produce NMDS. So this would have to be done with an R library. So the next thing we're talking about is interpretive methods. So these are statistical tests where we want to assess the plausibility of the proposed hypotheses. So once we've generated an ordination, going back here, how do we know that how can we tell whether these communities are different or not? I mean, just in general, we can say, well, yeah, I mean, the green samples look to be higher, look to be somewhat different in general from the red samples. But we need a quantitative way to test these hypotheses. So some of these kinds of questions: does the microbial community cluster according to predefined sample groups? Does the distribution of the community reflect underlying parameters? So chemical parameters, environmental data, these sorts of things. What component of the environmental data or patient data corresponds to the largest shift in the microbial community? These are all the kinds of questions we can ask with these sorts of methods. So the first thing we're going to talk about is multivariate tests of significance. There are three main ones that people use, and really the first two are the primary ones that people use today. But they all do different things. And again, this is the importance of understanding the tools that you're using. So ANISIM tests for significance between two or more classes of objects based on any dissimilarity metric, similarity metric. PERMANOVA, which is a more recent version, is a non-parametric method. So it's literally permutational ANOVA. It tests for differences between object classes again. The null hypothesis here is that the centroids, so the centroid is considered the center of mass of a distribution of objects. So the null hypothesis is that the centroids between the groups are all the same. And so the rejecting the null would be to say that there is a difference, a significant difference between the center of mass of each of the groups. The mantle test compares two distance matrices or two dissimilarity measures calculated for the same objects, but for two independent set of variables. So species distance matrix versus a site distance matrix. So that tends to be more used in ecological analyses where you have you're collecting samples from different sites. Again, I kind of did this for PERMANOVA already, but ANISIM, the null hypothesis is that the average ranks of the within group distances is greater than or equal to the average of the ranks of the between group distances. PERMANOVA is looking at group centroids, which is a little bit easier to visualize. And the null hypothesis for the mantle test is that there is no relationship between the interpoint distances in one distance matrix and that of a second distance matrix. So this is just a visual representation of ANISIM. So you start with a dissimilarity matrix that's similar to what we saw earlier on the left, and then it's transformed into a ranked dissimilarity matrix. And then the ANISIM statistic compares the mean of the ranked dissimilarities between groups to the mean of the ranked dissimilarity within the groups. So an R value close to 1 suggests dissimilarity between the groups, while an R value close to 0 suggests an even distribution of high and low ranks within and between the groups. And significance is then determined by permuting group membership a large number of times to obtain a null distribution of the R statistic. Canonical correlation analysis is a multivariate constrained ordination technique that attempts to extract major gradients among combinations of explanatory variables in a data set. Plain CCA isn't really useful because for microbial ecology because of the qualities of the data, but there are modern methods or modifications to CCA that are useful for microbial analysis. And so this is a correspondence plot where if you look at the angles between, if you draw lines from the center point to any two objects in the plot, if you have a small acute angle in the two lines between them, then those things are highly correlated. And if you have a large obtuse angle between them, then they're highly uncorrelated. And so this is an example of a more modern take on this is a correlation circle plot. Again, same idea. This is actually uh, from a multi-omic analysis. I believe this was this is an example from the Mixomics vignette, but this is correlating genes and I believe cytokines or something along those lines. And so again here, two things that you draw lines from the center to two things that have an acute angle, then they're highly correlated, and then the opposite is true. They're uncorrelated if there's a large angle between them. So again, these are a lot of tools. It's useful to have a good grounding in them. And so the last method, discriminatory methods, is to define what things discriminate, what parts, so whether it's functions or species or relationships among species, are most driving those patterns of interest. So what species are most responsible for separating the groups that you saw in the ordination plots, for example? So random forest, a random forest classifier, again, an oldie but goodie, we'll say. It's still useful, but with some modifications, it can be made more useful. Filtering, for example, mutual information filtering to remove highly correlated OTUs or species, for example, can improve the classification results. But the building block of a forest is a decision tree, which basically uses features from the data set and threshold values to build an ensemble set of trees. So it's the general idea that a bunch of weak classifiers combined together make a strong classifier. And so this random forests, these decision trees, will pick out a certain number of features, a small number of features that can discriminate between multiple groups, and then we combine multiple trees together, as you can see in the majority voting, to determine the final set of discriminant features between multiple classes. Networks or graphs, they're called, is another form of this. This is so the OTUs or organisms or whatever functions are nodes, and they're connected by vertices, and those vertices are relationships. How those relationships, whether they're correlations or, we'll see in a second here, Jacquard dissimilarity. So this is an example of using Jacquard dissimilarity with a threshold cutoff. So things that are highly similar to a certain degree remain connected by, and the strength that is connected by, or sorry, not strength because it's a presence absence, but if you set a cutoff of Jacquard dissimilarity, then you can see things that are groups of organisms that are connected by their similarity to each other. Okay, so that's the general overview. So we have, this is the third thing we have. So if we go back to the webinar, it looks like I did not include this. Okay, so I will go back and include, I have an R script going through a number of ecological statistic metrics, alpha diversity, beta diversity. I will go ahead and open that now. Apologies for not uploading that earlier, but you can follow along. So this, so this project, this was the data set for this was originally from the same Mothur SOP data, the mouse data, and I will paste into the the chat here, the workflow, microbiome workflow. So this is where the data with so I use some of this, I use the data from this, but and I use a few of the methods from this, like random forest, but it's a mix of this and some other analysis metrics that I'll be going through here. Okay, let me. Okay, so this is this Ecostats R file here. Just goes through again. Just a quick overview of all the stuff that I just talked about in interactive form. So these are the main libraries. Phyloseq for a lot of the alpha and beta diversity. Vegan for the statistical comparisons, the multivariate statistical comparisons. ggplot for plotting. And Mixomics for some of the advanced correlation analyses that I talked about earlier. We'll just kind of run through this real quick. This RDS file, I generated this separately and saved it as an R data file, so you don't have to go through and generate that. I will include this on the GitHub, so you can just start from here. So we can load this into a Phyloseq. This is a stored Phyloseq object representing a DADA2 run of the mouse gut data. So it's already pre-filtered and generated and everything. So we can just get started. We're creating a set of alpha diversity metrics here. This is just an array or a vector of metrics that we want to generate. And so we can pass those to the plot richness function that's part of Phyloseq. So, and we can combine it. So the plot richness generates a ggplot2 object that we can combine with standard ggplot2 geometries and other modifications. So this is just a basic plot of alpha diversity. We can see here for each of the 4 metrics we defined, observed species, Shannon diversity, Simpson diversity, and Fisher diversity. So we can go through and we're just going to add on to each of these. So instead of just a simple box plot, we can do violin plots colored by age. This is, again, the mouse age. Remember, this is from 0 days to 150 days. Or actually, I guess it goes for 200 days, technically. Oops, okay. Yeah, I need to make this a lot bigger so we can see what's going on. So a violin plot is basically a box plot that has been turned into a histogram on both sides. So if you look at it sideways, you can kind of see a histogram, or a smooth histogram. And on top of that, we've added a swarm plot. So you can actually, so there's multiple things here. So the box plots show you broad outlines of the data. The violin plot shows you more of the contours of the data. And then we add on top of that plotting of the actual data points in that swarm plot there. So all the black dots are the actual underlying data points represented by the histograms of the violin plot. So it just lets you see more of the actual data itself. And then since there are so many plot points there, it's hard to see. A lot of them are overlapping. We can add some jittering to separate those out. And we will take a look here. Again, this needs to be increased. So we've now colored the dots by their age bins. This is a bit ridiculous because we're coloring by day. This wouldn't necessarily be all that useful with that many different categories, but you can see the general idea. Again, showing differences in the same 4 alpha diversity metrics, and just showing some different variations in how we plot the points. In this case, we've added colors instead of just black. colors for all the points. We can see how those points are, the distribution of ages within those points. And so, to look for additional patterns to see if some of the, because we've got these bins here. So the young mice are from 0 to 100 days. Well, that covers 100 days. Is there any real variation in the alpha diversity metrics within that bin? So again, this is just showing you some, the different kinds of analyses you can do. This replicates the alpha diversity Kruskal-Wallis test that we saw in QIIME earlier. So we can do this with the estimate richness function, which is again part of Phyloseq. So for each of these, it produces the Kruskal-Wallis test of significance between the groups. And so between each of the groups, each of the 3 groups, there is a significant difference among all of the different observed metrics, or the different alpha diversity metrics. Let's see, run that here. So this is another example of normalization. I don't have time to show it here, but if you look at the, actually I guess I can plot it here. If you look at the log transform plot of the abundances, you see the log transform plot looks a lot more normal, looks fairly normal, indicating that the original underlying data was very not normal. And so we can use the log transform data to calculate some beta diversity metrics. Here we can calculate the weighted unifrac. So we calculate the metric, and then we can plot the ordination. So the ordination plot here by default, this is just a plotting function. So here we do this in 2 steps. So we calculate the distance metric, giving it the method weighted unifrac. We do the ordination. So ordination, give it the method of multidimensional metric multidimensional scaling, MDS, which is the same as PCOA that we talked about earlier, principal coordinates analysis. And then finally we plot the results. So we can see here, this is the plot we saw earlier in the presentation. Again, just the bins, the young in red, the middle-aged mice in green, and the older mice in blue. And so we can take that visualization. So we did weighted unifrac. We can also calculate Bray-Curtis, the other beta diversity metric method we mentioned. Again, we can do the ordination plot for that. Looks pretty similar. Bray-Curtis is showing a little bit more differentiation between them, between the groups. But as I said, visualization only gets you so far. So we can use the Adonis function in vegan to perform the PERMANOVA test. And so we will get an output in the console here. So grouping by the binned ages, the PERMANOVA test tells us that yes, in fact, there is a significant difference. Now, since this is a group-wise test, it doesn't tell us which of the groups are significantly different from each other. But as we saw, so we can do it with this. Yes. So Diana is asking, am I going to share this code? Yes. I thought I had included it when I uploaded everything to GitHub, and somehow I managed to leave it out. So I will do that as soon as the class is over. I'll go and upload this and the data set to GitHub so you can download this and run through all this on your own. So this will be available. One of the really great things about PERMANOVA that I didn't mention earlier is, and QIIME 2 implements this in a very nice way. They do it automatically for you. So if you run PERMANOVA on anything, sorry, I'm getting ahead of myself. So PERMANOVA can be used for group-wise significance, But if you get a significant result and the group-wise difference, then you can run PERMANOVA pairwise to get to do post hoc pairwise comparisons, just like you would do with two keys test or something like that. So you can run PERMANOVA once to get group-wise significance, and if you get a significant result, then you can run it pairwise to test, okay, now which of these groups are significantly different from each other. And the QIIME2 implementation of this does it automatically, and it produces their QZV, their visualization artifact for this. Makes a nice table with histograms, and it connects it to your metadata table, so you can go interactively and choose among the different metadata columns, and then it will automatically show you the group-wise PERMANOVA result as well as each of the pairwise PERMANOVA results, which actually, now that I remember, we actually showed that earlier in the slides. So yeah, so alpha diversity, beta diversity, ordination plotting, and PERMANOVA statistical testing. So the last thing that we will do here before we finish is to show the random forest. So we will generate a data matrix that can be used. So random forest is what's called a supervised learning method. You split your data into training data and test data. So the method learns the characteristics from the training data and then attempts to apply what it's learned to the test data. And if you get good results, and there's things you can do like cross-validation to make sure you're not over-learning from that particular subset of training data. But that's beyond the context, beyond the scope here. But just remember, you supervised training data, test data. So we split that up into training and test data. And again, this part in particular is gone into in more detail in that workflow I pasted earlier. I'm just showing some examples here. So we can actually, the train method here, you give it the method of random forest. There's other methods you can use, but we're training random forest here. And the end results, oops, let's see, what did I do there? Yeah, okay, let's run this again. Oh, because I did not load these libraries. That would be why. Okay, so let's try that again. Let's run the training. So this will take a few seconds here to train the random forest classifier on. So it's using age as the primary grouping that it's going to be discriminating as the objective function. So age is what it's trying to, is what the classifier is trying to discriminate amongst. And so we've got the three different bins of age that it's attempting to solve for, essentially. So once we do the training on the training data, then we will use the learned model to predict the classes of the samples that were reserved for the testing data. And then the third line in line 90 here is we will see the results of that prediction and see how good our random forest classifier was. Okay. And as soon as this is finishing, so we'll see just a simple 2x2 table. The table that we're going to see here will just show that 2x2 table of expected class and predicted class. And then in the second set of results here, in the plot results section here, we'll actually use the random forest classifier to do a sort of ordination plot, MDS plot associating the classified labels. So using the classified labels to create an MDS plot from the random forest data set. There we go. Okay, so that's finished running. So we finished the training, and then we can test out the training by predicting the new data set, or the test data, and then we can see here's the table. So this is what we want to see. So of the 0 to 100, so the young mice that were supposed to be young mice, it predicted 67 and only mispredicted 2. And same for the older mice. So the older mice were mostly predicted to be correct, and only 11 were misclassified as being younger mice when they were in fact older mice. So that's decent results. So we can go ahead and plot that, like I said, with PCOA, those results, and see how that works. So again, pretty decent classification results. And the last thing that we can do is check which taxa are actually driving those differences. Which taxa is the random forest classifier kind of grabbing onto to differentiate those classes. And this is just showing the two most important things. And so we're seeing Lachnospiraceae and Roseburia genera family and genera. So Lachnospiraceae, the family, and Roseburia, the genus, are the two most important discriminating factors between the two groups. And then we can just show a plot of the overall set of which classes are discriminated, which of the two classifiers, the predicted, sorry, how many OTUs are the classifiers grabbing on to discriminate between the two groups. And so in the younger samples, there's a very small number of OTUs that it's pulling to discriminate, whereas the older sample it's using less abundant but more organisms it's using to discriminate. So again, just a quick overview. That is it for everything. And we've gone much longer than I expected. So thank you to all of you who were able to stay.